Hello students, in today's class we are going to learn about nitrogen fixation process. Nitrogen fixation is basically the process of converting nitrogen into ammonia, which is then incorporated into amino acids and used for the cellular nitrogen requirement. The nitrogen fixing bacteria can be either free living or symbiotic. The process can be carried out in aerobic or anaerobic conditions. However, the enzyme nitrogenase is oxygen sensitive. So even in aerobic bacteria, it needs to be protected from exposure to oxygen. So the process is carried out by enzyme nitrogenase, which is made up of two different subunits, which are known as dinitrogenase and dinitrogenase reductase. Dinitrogenase subunit contains iron and molybdenum as cofactors in the form of a FeMO cofactor. So the actual reduction process of nitrogen will occur within the iron molybdenum center. Two copies of FeMO cofactor are present per molecule, and the molecular weight of dinitrogenase is 2,20,000 Daltons, while dinitrogenase reductase contains iron and has 4 iron per protein molecule. Molecular weight of dinitrogenase reductase is about 64,000 Dalton. So the process of nitrogen fixation is reductive in nature. That means it needs to be protected from exposure to oxygen, since oxygen can rapidly and irreversibly degrade the enzyme dinitrogenase reductase. So the enzyme must be protected from exposure to oxygen. So there are various mechanisms that the cell adapt in order to remove the oxygen. Either it will have a rapid respiration process so that the effective oxygen concentration becomes low, or it produces and secretes some oxygen retarding slime layer, or in case of heterocyst, the nitrogen fixation process takes place within the heterocyst structure, which are specialized cells. They have a very thick wall that will prevent the effective diffusion of oxygen. So by one or the other mechanisms, it will prevent the enzyme from exposure to oxygen. Coming to the biochemistry of nitrogen fixation process, the electrons for the reduction of nitrogen are transferred by dinitrogenase reductase, are further transferred to dinitrogenase. And here the flavodoxin or ferredoxin molecules will, reduced form of flavodoxin or ferredoxin will provide the required electrons for the reduction process. ATP also is required for the nitrogen fixation process. So during each cycle of electron transfer, the dinitrogenase reductase is reduced by either flavodoxin or ferredoxin molecules, and then it binds to two molecules of ATP. ATP binding changes the conformation of dinitrogenase reductase such that there is increased affinity of the subunit for the dinitrogenase subunit. As a result, the two will be associated with each other. In the process, dinitrogenase reductase will transfer the electrons onto dinitrogenase, and there will be simultaneous ATP hydrolysis. Hence, once the ATP is hydrolyzed, the association of dinitrogenase reductase for the dinitrogenase component decreases, and the two will dissociate from each other. And then the reduced form of dinitrogenase will carry out the last step, that is reduction of nitrogen to ammonia. So during this reduction process of nitrogen to ammonia, 3 cycles of one electron pair transfer will be required. For each electron transfer, 2 molecules of ATP are required. So overall 12 ATPs are required for a total of 6 electron transfer that is actually required for nitrogen conversion to ammonia. However, during this process, one molecule of hydrogen, which I have not represented here, it is also produced as a byproduct of nitrogen fixation process. And generation of one molecule of hydrogen will require 2 electrons, and that accounts for 4 more ATPs. So overall, you can say that 8 electrons are required during the reduction process of nitrogen to ammonia, which accounts for 16 ATP molecules. So here is the overall reaction during the reduction. And as you can see, one molecule of hydrogen is synthesized initially itself during the reduction of nitrogen to the NH level, and then finally it is reduced to NH3 or ammonia form. Overall, 8 electrons and 16 ATP are required for the reduction process. So this slide represents about how the distribution of work takes place in non-symbiotic nitrogen fixing bacteria. So non-symbiotic nitrogen fixing bacteria, here we are looking at the example of cyanobacteria. These are filamentous bacteria, and they have different types of cells that are present within the cell structure. So one type of cells are the vegetative cells. These are photosynthetic in nature, and they will carry out photosynthesis and generate sugars during the process. While the heterocysts are specialized only in nitrogen fixation process, they basically have thickened cell walls and glycolipids in their membrane that is going to decrease oxygen diffusion in the heterocyst cells, which is the required condition to protect the enzyme nitrogenase from exposure to oxygen. So nitrogenase is required for conversion of nitrogen to ammonia, which is incorporated into amino acid glutamine. Glutamine will then diffuse into the adjacent vegetative cells, and the vegetative cells will use up the glutamine to synthesize other amino acids by extracting the ammonia from it. And in return, the vegetative cells are going to provide the required sugars for the fixation process. Symbiotic nitrogen fixing process is important as it improves the soil fertility. Here we are going to look at the example of Rhizobium leguminacearum, which forms a symbiotic association with the nodules of leguminous plants like the pea plant. So the nitrogen fixation process is carried out by the bacteria in a nodule-like structures, which are known as nodules, and they consist of plant cell as well as bacteria. And because of the formation of these nodules, oxygen diffusion decreases within the nodule-like structures. In addition, the bacteria will synthesize a protein which is known as leg hemoglobin. It is a red color iron-containing protein. It is similar to hemoglobin of humans, and they are synthesized and secreted by the bacteria. These basically bind with oxygen in the nodules, and they will decrease the effective oxygen concentration by keeping low levels of free oxygen in the nodules. Thus, they are preventing it from degrading enzyme nitrogenase. This slide talks about what are the different stages of root nodule formation. The first step is recognition and attachment. Here, first of all, secretion of certain organic compounds by the plant cell plays an important role in stimulating the growth of rhizosphere bacteria. Once we have enough bacteria present in the soil ecosystem, then the root hair cells will have certain calcium complexes present on their surface, which will bind with ricadicin protein that is synthesized and are present on the bacterial cell surface. So interaction between the complex calcium complexes and ricadicin can be involved in initial recognition. Alternatively, certain sugars are also synthesized and secreted by the plant cell, and the bacteria make certain sugar binding proteins which are known as lectin. So lectin sugar interaction can also be involved in initial bacterial attachment. Once the bacteria have attached to the surface of the root hair cells, the second stage is penetration inside the root hairs. So bacteria will basically synthesize and secrete certain factors which are known as nod factors, which are basically plant growth promoting hormones such as auxins and cytokinins, and they will secrete it, so that will lead to irregular cell division in the root hair cells. That will result in curling of the root hairs, and during this process it will wrap up a group of bacterial cells, after which the bacteria can get access into the root hair cells. So once they have entered into root hair cells, they undergo a process which is known as infection thread formation, in which the bacteria will basically divide in inside a cellulosic tube that is formed, and the cellulosic tube will allow its access to the root cells. Now, once it has reached the root cells, then the bacteria will further secrete a large amount of Nod factors, and which will allow the rapid cell division of the plant cells, uh, which will result in formation of a nodule or what do you say, an irregular mass of cells that is formed, that is basically known as nodules. Now, once in the nodules, the nitrogen fixation process will form, and before the process, basically bacteria are transformed into structures which are known as bacteroids, whereby bacteria multiply rapidly within the plant cell, and they are transformed into swollen and branched forms which are known as bacteroids. And also, bacteroids become surrounded by portions of the plant cell membrane, and the association is sometimes also known as symbiosome. And only after formation of these symbiosomes, the actual nitrogen fixation process will begin. So coming to the biochemistry of nitrogen fixation process, um, the enzyme nitrogenase that is required for nitrogen fixation process is oxygen sensitive, and it is localized within the bacteroids, while the actual process of nitrogen fixation will take place. So the plant cells, which are photosynthetic in nature, they will provide the required sugars for that will provide the required amount of energy and electrons for the process. So these sugars will diffuse into the bacteroids, and here they will enter into the TCA cycle, will generate reducing equivalents, and that will provide the electrons required for the aerobic electron transport chain. So ATP is important for, as it is requirement for enzyme nitrogenase to carry out the nitrogen fixation process. So these electrons will go through the aerobic electron transport chain that will ultimately reduce oxygen to water. However, the main challenge over here is having oxygen within the same cell where enzyme nitrogenase is present, and it is important to prevent exposure of nitrogenase to oxygen. So here leg hemoglobin plays an important role. So no free form of oxygen is present within the cell, but leg hemoglobin molecule binds with oxygen, and bound form of oxygen will enter inside the cell, and only during the reduction step, as the reduction of oxygen to water takes place, it is released from leg hemoglobin. So in this way, free form of oxygen is not present in the cell, thus preventing enzyme nitrogenase. Then the pyruvate that is also reformed from malate it provides the required electrons for reduction of nitrogen to ammonia. Ammonia that is generated is incorporated into amino acids, and it will provide the required amino acids for the bacteria as well as the plant cell. So that's it about nitrogen fixation. I would request you to subscribe to the video so that you will get, you can just browse through the channel for the uploaded videos. Thank you.